Dear audience, after studying the properties of tissues during our introductory lecture, today we will consider the mechanisms of tissue formation at the main stages of embryonic development. The totality of these processes is denoted by the term embryonic histogenesis. Embryonic development, or embryogenesis, is the stage of individual organism development from the moment of fertilization to the completion of the main organogenesis processes. Embryogenesis is the most extensive period of life of any multicellular organism in both quantitative and qualitative terms. It can be distinguished by three main features. Firstly, it involves the intensive growth of the organism and its parts. For example, during human intrauterine development, the mass of the original zygote increases by no less than 1 billion times. The second characteristic feature of embryogenesis is the progressive growth of cell diversity, ultimately leading to the formation of about 250 cell types within 60 main tissue varieties. The examination of this phenomenon is the main topic of our lecture. And the third most important feature of embryogenesis is the formation of the complex external and internal body configuration of the embryo. This process referred to as morphogenesis. Embryogenesis includes a series of stages that are universal for all multicellular organisms. The existence of any organism begins as a result of fertilization. Fertilization presents the fusion of male and female germ cells. Following penetration of sperm genetic material into the egg cell, a new unicellular living system occurs called a zygote with diploid chromosome content. The process of development of zygote into a multicellular embryo formed by a single germ layer, blastula, is designated by the term cleavage. The different cells derived from cleavage are called blastomers. After cleavage, gastrulation follows. Gastrulation is the stage in the early embryonic development during which the blastula is reorganized into a three-layered embryo known as the gastrula. The result of the differentiation of the germ layers is the formation of the axial complex of germ organs. The first axial organs of the embryo are the notochord and the nervous tube. And the final stage of embryonic development is histogenesis and organogenesis. This period is characterized by active formation of definitive tissues and organs. Histogenesis represents the process of forming various tissues through the differentiation of cells as a result of implementing their genetic developmental programs. Organogenesis, in turn, signifies the development of organs from tissues. At this stage, specialization and organization of tissues into organs with specific functions occur. It is important to note that in humans, this is the most prolonged period, occupying 31 out of 39 weeks of intrauterine organism development. Fertilization is the process of merging male and female haploid sex cells into a qualitatively new formation, a zygote, representing a single-cell diploid organism. Fertilization is at the core of sexual reproduction and ensures the transmission of hereditary traits from parents to offspring. The interaction of the ovum with the sperm is a multi-stage process and includes contact interactions of gametes, penetration of the sperm into the ovum, and the formation of the zygote. The final phase involves several stages. The sperm, having penetrated the egg cell, forms the male pronucleus. The two pronuclei, male and female, come together and their fusion occurs at the syngamy stage. In humans, the total duration of the process from the beginning of the interaction between the egg cell and the sperm to the first cleavage is approximately 30 hours. The dynamics of the fertilization process in humans under in vitro laboratory conditions are shown in the video clip. From the perspective of the concept of a clone as a set of descendants of one stem cell, the human organism is a clone of the zygote. The zygote is characterized by totipotency, the ability to give rise to the whole diversity of cells and tissues of the future organism. All cells of the developing embryo receive the same set of genes, but differ from each other in their morphology, biochemical, and functional properties. These differences progressively increase during embryogenesis. At the core of this phenomenon lies the functioning of different genes in the embryo's various cell types. At each stage of embryogenesis, the majority of genes are in an inactive state, and only up to 10% of genes are actively functioning. Initially, genome regions responsible for the common properties of cells are activated. At later stages, genes encoding the synthesis of specific products that determine the phenotypic characteristics of cells are activated. The combination of the above described molecular mechanisms is referred to as determination. Determination is the programming of cell development based on the selective expression and repression of genes. The initial stage of determination processes is the differentiation of the zygote cytoplasm. The differentiation of the zygote cytoplasm, or ootypic differentiation, begins during the fertilization process. As a result of ovogenesis, the oocyte accumulates substances that provide the early stages of embryonic development. Firstly, this includes reserves of nutrients and energy resources, known as yolk. The second important component of egg cells is morphogenetic factors, or cytoplasmic morphogens. Morphogenetic factors are transcription factors that regulate the activity of zygote genes and guide cell differentiation in different directions. The entry of the sperm sharply activates the metabolism of the egg cell. The trajectory of sperm entry into the egg cell leads to the redistribution of morphogens and nutrient reserves in the cell, thereby determining the increasing heterogeneity of blastomas during cleavage. The figure shows that already at the stage of 4 blastomas, the sets of morphogens in neighboring cells differ significantly. Heterogeneous blastomas possessing different cytoplasmic heredity further differentiate into various types of cells within the embryo. Cleavage is the second stage of embryogenesis. Cleavage presents a series of repeated mitotic divisions of the zygote and its daughter cells, which are blastomas or cleavage cell. This process has a number of specific features. Cleavage is characterized by a high rate of division because the duration of blastoma interphase is much shorter than that of somatic cell division. The G1 period, during which the cell grows back to maternal size after mitosis, is practically absent. So with each division, the daughter cells decrease in size. As a result, the size of the embryo does not change during cleavage. The blastomas do not separate and, through intercellular contacts, form the final structure, the blastula. In the video clip, using accelerated filming, the process of human zygote cleavage under laboratory conditions is shown, resulting in the formation of a blastula in the form of a bubble or blastocyst. During the cleavage, differently determined blastomas form groups of cells with similar properties, which are the primary primordia of future tissues. In all chordates, 6 universal types of primary primordia can be distinguished. These include neuroectoderm, ectoderm, precaudal plate, notochord, mesoderm, and endoderm. Determination or the programming of the subsequent developmental path of blastomas at this stage of embryogenesis is not yet final, so the further fate of the cells developed from them can only be discussed with a certain degree of probability. The distribution pattern of blastula wall primary primordia and the dynamics of their movement in subsequent stages of embryogenesis have been experimentally studied using in vivo labeling of embryo regions with colorful vital dyes. Gastrulation is a complex process of cell reproduction, growth, directional movement, and differentiation, leading to the formation of 3 embryonic germ layers. The embryonic germ layers are anatomically distinct structures of the embryo that form during gastrulation and give rise to various tissues. Gastrulation is characterized by various cellular processes. Cell proliferation continues with varying intensity in different parts of the embryo. The most characteristic feature of gastrulation is the movement of cell masses within the blastoderm or in separate groups. Cells are sorted according to their belonging to different embryonic germ layers within which cells recognize each other. In doing so, some parts of the embryo can induce the formation of other structures. This influence of one structure in the formation of another structure is called embryonic induction. During gastrulation, 3 embryonic germ layers are formed: the outer layer or epiblast, the middle layer, and the inner layer or hypoblast. Gastrulation proceeds in 2 stages. In the first stage, 2 embryonic germ layers, the epiblast and hypoblast, are formed. In the second stage, the middle embryonic germ layer is formed between them. It is important to note that the formation of 3 embryonic germ layers as a result of gastrulation is a universal regularity in the embryogenesis of all chordates. During gastrulation, along with the movement of cell masses, the properties of cells in the primary primordia inevitably change. At this stage of embryogenesis, the programming of the cell development path in the primordia becomes irreversible. Therefore, upon completion of gastrulation, we have every reason to speak of the formation of definitive or embryonic primordia. Embryonic primordia are groups of cells systematically distributed in the wall of the gastrula, irreversibly determined to develop specific tissues of the new organism. After gastrulation, embryonic primordia are systematically distributed among the 3 germ layers. The outer layer, epiblast, is formed by ectoderm and neuroectoderm. The middle layer consists of notochord and mesoderm. Within the inner layer, hypoblast, there is material of endoderm and precaudal plate. The subsequent manifestation of cell differentiation in the embryonic germ layers is the formation of the axial complex of germ organs. This is the initial step of the histogenesis and organogenesis stage. After the formation of the 3 germ layers, the cells composing them interact with each other and induce the formation of the first axial structures of the embryo, the notochord and neural tube. The processes of their formation are referred to as neurulation. The primary organizer initiating neurulation is the cells of the notochord primordium. The notochord develops from the central region of the middle layer and serves as the first anatomical axis of the embryo. As a result of embryonic induction by the notochord primordium on the dorsal side of the embryo, thickening of the central region of the ethoblast begins, and neuroectoderm cells form the neural plate. Its edges thicken and move upward, forming folds or neural ridges, which eventually fuse to form the neural tube. The dynamics of neurulation processes are depicted in an animated video clip. Simultaneously with the formation of the neural tube, the mesoderm differentiates into dorsal and ventral parts. Growing dorsal mesoderm becomes segmented and forms paired round somates, lying along the notochord. Each somite consists of 3 parts: external dermatum, intermediate myotum, and internal sclerotum. Derma of the skin arises from dermatum. Myotum gives rise for skeletal muscular tissue. Sclerotum is source of bone and cartilage tissues. The ventral mesoderm does not segment and forms the splanchnoderm. It includes visceral and parietal layers as well as the secondary body cavity located between them, the coelon. The layers of splanchnoderm give rise to mesothelium, striated cardiac tissue, adrenal gland cortex. Each somite is connected to the splanchnoderm by a segmental stalk called the nephroderm. It gives rise to epithelium of kidney and reproductive organs. The division of the dorsal mesoderm into individual somates begins in the cranial regions of the embryo. In humans, mesoderm differentiation is completed with the formation of 42 to 44 pairs of somates. Except the dense mesoderm, there is a rather loose portion, which lies everywhere between germ layers and axial organs, and it's called an embryonic mesenchyme. Small mesenchymal cells with their processes form a meshwork and participate in the nutrition of all embryonic structures. Embryonic mesenchyme gives rise to the fibrous connective tissues, smooth muscle tissue, blood, lymph, hematopoietic organs, blood and lymph vessels. Alongside the formation of tissues contributing to the construction of the embryo's body, a portion of the cellular material of the germ layers differentiates into tissues of provisional organs. This term refers to temporary organs that function only during the embryonic period. They may perform functions specific to the embryo or basic functions of the organism until the formation of similar final organs peculiar to the adult organism. In a histological specimen of a developing chicken embryo, the external regions of the ectoderm, mesoderm, and endoderm, separated from the embryo's body by body folds, serve as sources of provisional tissues. Extraembryonic cellular material contributes to the formation of the yolk sac. Onion, allantois, serous membrane, or chorion. Due to the limited lifespan of provisional tissues determined by the duration of embryonic development, their histogenesis is characterized by early formation, accelerated differentiation, and rapid aging. The stages of tissue formation during embryogenesis are depicted in an integrated scheme. Cell differentiation directions begin to be programmed at the zygote stage, are determined at the blastula stage, and are finally established in embryonic primordia at the end of gastrulation. At this time, sets of stem cells for the main tissue groups are formed in each germ layer. During histogenesis, populations of stem cells for each tissue are formed from the stem cells of the germ layers. As a result of their subsequent differentiation, the complete set of tissues of the developing organism is formed. After birth, they continue their development. The processes of forming mature tissues are referred to as post-embryonic histogenesis. At the conclusion of the lecture, to obtain a comprehensive understanding of the process of embryonic development, I suggest watching an animated video film dedicated to the development of the chicken embryo, a classic subject of experimental embryology. Dear students, in this lecture, we focused in detail only on one aspect of embryogenesis, studying the mechanisms of tissue formation as an introduction to general histology. However, your acquaintance with embryology, the science of embryo development at the Department of Histology, does not end there. Upon completion of the course in special histology and the study of the male and female reproductive systems, the final section of our educational program will be a special lectures and practical sessions dedicated to human embryonic development. Stay with us for our next meetings on the HistoNavigator channel.